Hello, in this video we will discuss about the transcription and translation. We will just understand about the overview of the transcription and translation. So here is the chromosome, basically the 46th chromosome in human cell, which that is the diploid. Half is come from the mother and half is come from the father. So here is the nucleus. Basically here is the nuclear membrane. So remember about that. Here is the nuclear membrane which contain channel, a nuclear pore. So let's begin to understand. Here is the chromosome is contain DNA as well as the histone protein. Histone protein is the 60% while the 40% is the DNA, which that is wrapping around the histone protein. Four type of histone protein contain 8 subunit, and linker protein is also used for the linking to each other and condense formation. So during the DNA is the helicase enzyme which create the DNA double helical structure to unwind, and unwinding to create a DNA bubble. So this DNA bubble is very important. And here is the promoter region, and here is the coding strand, which that is the basically coding, and here is the template strand, which that where the messenger RNA will be transcribed. So here is the messenger RNA will transcribe from 5' to 3' direction, while here is the template DNA is the 3' to 5', not 5' to 3'. That's mistake. So here is the last is the stop signaling signal series is the adenine, thymine to guanine to cytosine. So the RNA DNA hybrid helix will be formed. So this is the template strand. So let's begin to understand. When after the transcription, the stop signal or termination will begin and produce the messenger RNA. This is the messenger RNA is known as the pre-messenger RNA. This pre-messenger RNA contain exon and intron. Exon contain coding sequence, while the intron is the non-coding sequence, which that is restriction of intron, and it means the intron will be released after the modification, and pre-messenger RNA will become a messenger RNA. This pre-messenger RNA will become a 7-methyl GTP containing cap and poly A tail. This poly A tail and 7-methyl guanosine triphosphate attachment is the cap is the 5' site, while the 3' site is the poly A tail. What is the benefit of this cap and tail? The cap and tail is used for the protection into the cytoplasm from the nucleases and phosphatases enzyme, which that is protect from the damaging of the messenger RNA during the export. So here is the basically the polymerase 2 is binds on promoter and template strand, and 75 to 25 sequence is the binding of the eukaryotes. So the RNA polymerase 1 is used for the ribosomal RNA, while the RNA polymerase 2 is used for the messenger RNA transcribe, and RNA polymerase 3 is used for the transfer RNA. So remember about that. So let's begin to understand also the transfer RNA and ribosomal RNA. So the first of all we will discuss about the here is the exportin, which that is the export of the protein, export of the messenger RNA by a channel is known as the transporter is known as the exportin protein. While here is the nucleolus. Nucleolus is a sub part of the nucleus which that contain also DNA. This DNA is the, for example, the ribosomal DNA cluster. This ribosomal DNA cluster will be transcribed just like a transcription of messenger RNA, but the RNA polymerase 1 enzyme is used for the transcription to produce 47 pre-ribosomal RNA, which that is bound with the NCL1 and NPM1, which that is protein splicing of the segment ATS, 5.8S and 28S processing, and after this assembly will be formed, and this assembly will become a pre-60S and pre-40S ribosome subunits. So basically 60s and pre-40s is the immature still. So after this, for maturation, is the ribosomal protein for large subunit and small subunit is enter into the nucleus when the ribosome will go into the outside of the nucleolus. So it will be move inside, and this RBL and RBS, which that is a ribosomal protein for large subunit and ribosomal protein for small subunit, will be add. After this addition, basically is the uh, this ribosomal RNA will be add the protein. So it means the protein is less, but the ribosome contain RNA containing content is higher as compared to the protein. So after this, exportin will be export this ribosome outside. So let's begin to understand also nucleolus will also release the transfer RNA by the transfer RNA cluster genes. So here I am not drawing the transfer RNA cluster genes. So the here is the pre-transfer RNA will be formed, which contain a loop-like structure P site and D site. After this cutting, the P cut and D cut from the hydroxyl site and 5' site and 3' site. After the cutting, that is a cleavage formation, and the cleavage will be basically is the splicing and cleavage to make a basically releasing of the P site and releasing of the D site, and addition of cytosine adenine to 3' at end will be addition by the enzyme, which that is used for the enzyme, and this enzyme is the mature transfer RNA will be formed. So after this mature transfer RNA, which contain a ACC and hydroxyl group with the 3' end, while the 5' end is not. So here is the 3 loop like structure. So the down loop contain a anticodon. So remember about that. This anticodon will be bind with the codon of the messenger RNA. So when will bind with the messenger RNA, so the protein containing transfer RNA will shift to another polypeptide, another amino acid to bind. So here is first of all we will discuss about the amino acyl transfer RNA synthetase for methionine. For example, the first amino acid will be bind and transcription will begin. So here is the transfer RNA will be bind with this enzyme with the ATP. So ATP will be bind here, and the methionine amino acid will be bind here, and it's bind due to the hydroxyl group. So the hydroxyl group will be interact with the amino acid to bind it. So that is why basically is the H2O water will be released, and it will become ATP will convert into the pyrophosphate and AMP. So the adenosine monophosphate conversion, and due to the 2-phosphate group will be released, it means the high energy required. So I hope you make sense for the attachment of the amino acyl transfer RNA synthetase for methionine binding, for example, so the amino acid. And next is we will discuss about after the shifting of the amino acid to the transfer RNA, for example the methionine. So the amino acyl transfer RNA synthetase for methionine will be released, or amino acyl transfer RNA synthetase will be released, while the AMP plus pyrophosphate will be released, and it produce the transfer RNA, active transfer RNA which containing methionine. So that means basically is the reduction of the methionine with the transfer RNA. So the anticodon are present. For example, the methionine is the initiation of the translation. So this is the translation mechanism. That was the transcription, but here is the translation. So here the ATS ribosome will be formed when bind with the help of magnesium. So the 5-methyl 7 here is the, you can see in here, and I am drawing the bigger picture to understand A side, P side and E side. So the A side, basically when A side will be add the transfer RNA with the codon AUG, and this codon is the anticodon is the AUG. Basically AUG is the codon which that is contain a UAC, so that is the UAC containing anticodon. So the messenger RNA will move here while the transfer RNA will move like here, like this manner. Codon will be moved. So you can see in this diagram to understand. It means the messenger RNA will move left while the methionine bind with another protein is right side. So you can see after the further process. So here is the another transfer RNA will be bind with the another codon with anticodon. So it means here is the number 2, which that is you can see here the methionine containing transfer RNA will be here, and after the here is the second one diagram to understand. Basically I am drawing just one ribosomal subunit. It's contain basically. So here is the methionine will be shift in the peptidyl side, the P side, and addition side will be add more transfer RNA. So after this peptidyl side will be this amino acid will be attached with the peptidyl side and release the from the A side to move P side. After the P side, it means when E side, the methionine containing transfer RNA will give the methionine to the P side containing transfer RNA. So it means here you can see that is the new will be added, and here is the releasing or exit side. Exit side will be exit, and the another will be add here, and to the synthesis of the polypeptide will be formed. This synthesis of the polypeptide, basically the polypeptide is due to the P side, and A side is the addition side, 1 and 1, one add and one exit. So it means the P side remain constant and change the 1 by 1 to add more. So E side, when go E side, so it will be transfer the amino acid to the P side and due to the peptidyl side. So after this, the UAG, UGA, UAA basically is the terminal codon, and here the polysomal ribosome formation. So that thanks for watching.